Okay, so maybe we can get started. So welcome everyone to this webinar. This is part of a webinar series on generative AI and statistics that's been run by the Stats Up AI Alliance. So I'll just talk very briefly about that. So the Stats Up AI group is a new group that kind of aims to organize activities to empower statisticians to participate more actively in AI research and leadership. And it's been recognized recently as an ASA interest group. And if you're interested in finding out more information, you can check out our website here. And we do have several webinar series going on at the moment, and the recordings of these webinars are available on our YouTube channel. And so today we're very fortunate to have Dr. James Zou speaking. James is an associate professor of biomedical data science at Stanford, and where he leads the Stanford AI for Science Lab. And he's won numerous awards throughout his career, including the Overton Prize, an NSF Career Award, a Sloan Fellowship, and many more awards here that I won't list. And James is working on making AI agents more capable and reliable for important applications in healthcare, biology, chemistry, and medicine. And so James, the title of James's talk today is AI Agents to Accelerate Scientific Discoveries. And James, it's all yours.
Great. Thanks so much, Owen. Shared my screen. Can you see my slides?
Yep. And sorry,
Okay.
just before you start, James, just a quick announcement. So James mentioned there will be a few points where he'll pause for questions, but otherwise we can maybe defer some larger questions to the end. Sorry.
Okay. Sounds good. Yeah. And if you also have questions, you can just feel free to raise your hand. Great. I'd love for this to be very interactive. Great. Well, yeah, so it's really my pleasure to participate in this webinar. And I want to share some of the directions we're exploring recently around developing AI agents to help us to accelerate science. So the broader context of this is that I think we're in this quite interesting conceptual shift moment from thinking about AI as a tool for science to think of AI as scientists by itself. Right. So for the past, decades, right? So AI has been very successfully used as a tool for tackling specific scientific problems. And typically, researchers would come up with like a well-designed problem, like maybe you want to predict structures of proteins. You can collect the data, and then you can apply or train an AI model for solving that specific task, right? In that case, the AlphaFold will be an example of a very useful tool for science. Over the past year in particular, right, the paradigm shift is that, right, rather than having AI as a tool for tackling a narrowly defined problem, we're starting to think about AI as a co-scientist, where this AI can by itself come up with research ideas, create its own tools, analyzing data, and maybe even helping us to write papers. Right. So in that sense, then these AI then becomes not simply tools, but really more like scientists that can help to accelerate scientific discoveries across the much broader and more flexible spectrum. So that change and that advance is largely driven by AI agents, which for our purpose, we can think of them, these AI agents as basically systems that we can build on top of large language models, right? Where the large language model, like your favorite GPT or Claude models, can sort of be the brain of that system. But then we also give these systems additional tools, right? Also additional resources like external memories, access to different databases, research papers. So this enables the AI agents then to become much more flexible and more versatile in tackling a broader range of research endeavors. So I think it's very exciting. It's still in an emerging stage. So what I want to do today is to give a few examples to illustrate how we create these AI scientist agents and illustrate the kinds of scientific discoveries that these agents can make. And I'll also try to highlight some of the limitations and complementarities of how humans can best collaborate and work together with these AI agents. Great. So first, I just want to give a concrete example to illustrate the kinds of scientist agents that we can build and also the kinds of science that they could do help us to accelerate. So we, over the last year, developed this platform that we call the Virtual Lab, which is sort of a team of different scientist agents that work together. So we created the virtual lab to very much mirror my physical lab at Stanford. So in the virtual lab, there's also an AI professor agent. And then working with the AI professor agent, there's actually a bunch of different AI student agents with different expertise, let's say in data science, in machine learning, computational biology, immunology, and so on. And these AI agents, they have their own group meetings, and we give them a budget so they can actually perform and carry out some experiments. We also built for the agents a school, like a replica of Stanford, where the agents can actually go to the school and then teach themselves on how to become a better researcher at their specific domains. So this was published and described in more detail in this paper. So I'll just want to explain a bit more of how this agent virtual lab works. So the way that we interact with the agents in the virtual lab is that we can explain to the professor or the PI agent some project that we're interested in, right? So a project could be, I want to design a new kinds of proteins, or I want to analyze a complex data set, or design a clinical trial, right? So these could be all be different kinds of projects. And then the PI agent would look at that project and then say, okay, for that project, what different areas of expertise do I need to tackle that project, right? And then the PI agent has the flexibility to train and to create the sub-agents that have that expertise. So for one kinds of projects, maybe the PI agent might say it's useful to have an immunologist on the team. And then it's actually up to the PI agent and to create or train this immunologist agent. So in this way, the PI agent or the professor agent has the flexibility to actually create its own teams of experts and to adapt and customize that for different projects. So it's very versatile. I'll come back to and explain in a couple slides how that training and creation process of these expert agents look like. For now, you can think of each agent as taking a frontier language model, which could be, let's say, GPT-4 or GPT-5 models. And then we do additional fine tuning on top of these models to teach it how to become better at their specific areas of expertise. And once all these agents are automatically created, right, in the virtual lab, then they can start to have their own group meetings where all the agents will meet together to discuss and come up with a research plan. They can also have one-on-one meetings where one of the agents will meet with the PI and the manager agent and to review a specific subtask, which could be like going over a piece of code or analyzing a data set. Right? And they can alternate between group meetings and the many individual meetings. So to make it more concrete, I want to show you an example of one of these virtual lab group meetings. This is actually the first project that we assigned to the agents, which is to help us to design new binders to the latest SARS-CoV-2 variants. All right, so at the time of this project, there were some of these new COVID variants, and we didn't have great binders to those. So it's kind of an important, but also very timely research project, an open-ended project for the agents to help us to design these binders. So at the beginning of this project, the PI agent explains to all the other agents, right, what is the goal here? The goal is to develop binders, right, that can target the recent spike proteins, right, of these SARS-CoV-2. And then you can see the other agents can then speak up one after another to contribute their ideas. Right? In this case, the immunologist agent spoke up second and then recommended designing these binders in the form of nanobodies. And this is already a little bit surprising because nanobodies are these kind of sort of small cousins of antibodies, right? So they're relatively small proteins and they're only found in a few animals like camels. Most of the human researchers might have said, okay, let's design antibodies, which are much more widely used and more common. So it turns out that there's actually a good reason for this maybe unorthodox recommendation. So the machine learning agent spoke up after the immunologist and says that, I actually agree. I like the idea of designing nanobodies rather than the larger antibodies because nanobodies are smaller. That actually simplifies our jobs as agents, right, to computationally predict and model their structures. You can see how these agents can actually provide reasonings and rationales to support the recommendations. In all of these meetings, we also have a critic agent, sort of like a reviewer, that sits in on all of these meetings. Here, the goal of the critic agent is to provide more skeptical feedback, right, to the ideas of these other agents, right, to point out weaknesses, loopholes, and so on. Like here, the critic agent basically says that, well, we don't have a lot of data for nanobodies for COVID, right? So if you're going to go down that path, then we need to be very careful about overfitting these models. So the actual meeting for the virtual lab goes on for longer. They have multiple rounds of discussions. Here, I'm just showing the first part of the meeting, and you can already see how the agents can contribute their ideas, right? Build ideas off each other, and then relatively quickly converge into a concrete research plan. Now, one thing that I'm very envious of the AI scientists is that their meetings are much more efficient than my meetings. Right, so one of the virtual lab meetings, maybe it takes a few seconds or a minute. So in the time that I had one of my research discussions, right, they already had hundreds of meetings. And unlike me, the AI scientists, agents, they don't get tired, right? So they can do this research 24/7. And because they don't get tired, they actually also have multiple meetings in parallel. So for every question, right, the AI scientist wants to ask, they will actually discuss that 5 times in parallel. In each of these parallel meetings, there's some randomness, right, in the language models. They can come up with different ideas. And then the manager or the PI agent then will actually read over the transcripts of all these parallel meetings and sort of pick and choose the best ideas from these different meetings and then combine them into a concrete plan for the next steps. This is kind of an effective way to explore a broader range of ideas in parallel, right, to be more creative, right, and still have like a set of concrete action items. In case you're curious, I'm here showing in this pie chart basically how often the different agents would participate, right, in these virtual lab discussions for this project. So for example, the human researchers, like us, right, we only talk about 1% of the time, right? So we can participate in any time we want, but often we mostly try to provide more high-level guidance to the agents. For example, we tell the agents in the beginning, right, our goal here is to design binders to COVID variants because it's important to us humans. We also can give the agent some constraints, like how much money we have to do the wet lab experiments. Otherwise, we mostly let the agents actually come up with their own research plans, right? And then to implement and execute on their research ideas. So we don't want to like micromanage and tell the agents exactly what to do because we want to see how creative the agents are and come up with different ideas. And here the different agents have different roles, right? So for example, the computational biologist agent's role is to actually come up with the plan for how to design these nanobodies, right? Which I think makes sense so that these computational biologist agent actually has, does most of the talking, right? The machine learning agent's job is to actually do the coding, right, for the other agents. Like another agent might tell the machine learning agent, train this model or implement this algorithm, and then the machine learning agent will actually then go out and write the code for training these models. So they have different specializations. Great. Any questions so far about how the agents interact?
Yes, if I may. What do you find is the benefit of having these agents separated? Wouldn't it make as much sense to put all your investment into one massive model that, in a sense,
Yeah.
is to be subdivided? I yeah.
Yeah, so that's a good question. So we actually did that comparison in our paper, and we found that this multi-agent team to lead to much better solutions. And I think what happens is that when you have multiple agents with different backgrounds, so first is that they actually get into debates and disagreements with each other, right? And they have to resolve those disagreements. That actually process actually elicits more robust and more creative reasoning. Whereas if I have a single agent and try to do this project all by itself, no, that actually does not go into as in-depth reasoning as we have these multiple agents. And the second, we also found that it's actually also easier for training like teams of multiple agents, right? Each with more specialized expertise than training a big model together. And I'll tell you more about the training process through this virtual lab school in the next slide.
So maybe my question is about the training process, but basically to make these models talk to each other, you need much more than just the model, right? You need to give them some sense who should speak up when, how they should converge on a common ground. So I guess you'll talk about it, how to train these interactions, right? Or you need
That's right.
memory probably as well.
Yes. Yeah, so it's a good question. So for every agent, we also create like a external memory, sort of like a digital notebook, so they can remember all of their previous discussions and group meetings, right? And moreover, it's actually also the role of the principal investigator or the PI agent often to like organize these meetings, right? And so it's sort of like similar to how, for example, we might run our groups, right, where the professor might like say, okay, why don't you speak or why don't you do this task, right? So the PI agent here is also sort of like the manager that can help to organize all of these other sub-agents.
So there is a lot to be built, or do you see now that there are every day something new emerges? Pretty soon, I guess we'll have ready to go ways to specify these personas and let them interact. Or do you need to do a lot of custom work to make
Yeah,
it
so
happen?
we did build our own infrastructure for the virtual lab because we found that the existing available infrastructures are not flexible enough to accommodate these kinds of more flexible, open-ended scientific explorations. as a part of our paper, we actually released all of our infrastructure. It's all open source, right? So that other people are now using it and can easily adapt it to other problems.
Oh, thank you. So it will be great to see the paper.
Yes, yeah. So it's I think I put the link to the paper here, but this is the reference where we have all of the data code that's released.
Thanks.
Yeah,
Yes,
I have a quick question.
please.
Thank you. So do you have to set the rules of maybe conflict resolutions within the agents, or it's just something I was thinking about?
Yeah. Yeah, so it's a good question. So we don't do that ourselves manually, right? So we actually mostly leave it to the PI agent to decide how to resolve conflicts. And often what PI agent would do is that they will let these other agents, right, maybe discuss it a few more rounds, right? Try to see can they reach some consensus. And if they cannot reach a consensus, then often the PI agent might make like a decision itself, right? Says, okay, I've heard, both sides, and then I think, this is the best thing to do, and let's do the following.
Thank you.
James, I also had a quick question, if I may. So I'm sure that you have tried a lot of different settings of the meetings and all that. Is there any lessons that you learned that was generalizable in terms of what kind of setup work and what setup doesn't work?
Yeah, so we did find that actually the structures of having these parallel meetings is quite useful, right? And in particular, in each of these parallel meetings, you can also have different agents speak in different orders, right? So that ends up being quite important because, for example, when humans meet, right, so it depends on who asks the first question, that also, or who speaks first, right? That changes the trajectory of which ideas come up. Um, One thing that's I think is quite useful is that have the agents actually meet in parallel, right? And then each of the parallel meetings maybe change the order. Maybe in one meeting have the data scientist speak first. In the second meeting, let's say have the computer scientist speak first, or the third meeting have the the biologists speak, right? And then I think that actually can also create more diverse and more interesting ideas. Great.
May I also ask a question, James?
Please.
Okay, great. I wonder, in your experiments, I imagine you have done thousands of simulation studies. I wonder, have you encountered some situations where all those meetings or conversations ended up with some like fake or
Yeah.
irresponsible conclusions?
Yeah.
Because those are kinds of the situations I sometimes come into as a real human researcher.
Yeah. So that's also an important question. So sometimes we do find that agents can make mistakes. They can also be hallucinations. There are several guardrails that we build in that we think are actually quite useful. So first is that we actually have this critic agent, right? The job of the critic agent is to actually review all of these work that's done by the other agents, right? So it's actually very useful that the critic agent participates in all of these discussions and meetings. The second guardrail is that we also try to enforce all the agents do their work through coding, right? Because it's actually then much easier for us to audit and to run the code and check the code, right? And that we also reduces hallucinations. Finally, I think the most important check is that we actually do the wet lab experiments to then to validate all of the designs and findings from the agents. And that wet lab experiments sort of serve as like the ultimate ground truth that then provides feedback to the agents. And I'll share in a couple of slides the kind of experiments that we've done.
Thank you very much.
Great questions. So now that we see how the agents can interact, I want to come back to you and explain a little bit more of how the training process of this agent looks like through this virtual lab school. The motivation for having school for the agent is that if you take these frontier models, right, like GPT-5 or Anthropic, these models are very good at broad general knowledge. But if you go very deep into a specific technical scientific domain, these models may not have the most up-to-date or the sophisticated reasoning, right, in that technical domain. So to fill in that gap, that's why we built this agent school. And the way that the agent school works is that first, the humans, we can tell the agents some of the topics that are important for them to learn about. For example, one topic could be, learn about all the latest SARS-CoV variants, right? Or learn about the latest developments in cell therapies, or learn how to use AlphaFold, right? These are all could be topics. And then for each topic, right, the agent would actually generate their own learning curriculum by coming up with a set of questions that are relevant for that topic. And then based on those questions, we enable the agents to actually go out and search and find and download reference materials that are relevant for their topic. So for example, agents can go to PubMed Central, right, and then actually find and download open access articles that are relevant for each of these questions and topics. After they download these reference materials, right, then we enable the agents to start to read and to learn from those reference materials. So basically what happens under the hood, right, is that as the agent is actually reading and learning each of these materials, then we enable them to basically update the parameters of the neural network, the transformer that underlies these large language models. That helps the agents then to ingest that new information. So even if in the beginning, right, that multiple agents might start off as being the same base model, like a GPT-4 model, right, as they go through the school, right, so the immunologist is learning from one set of materials, the computer scientist is learning from a different set of materials, and then they're updating the model parameters, right, so then the agent starts to diverge, right, into different models. And we also have it won't be a very good school without some sort of testing, right? So we also have an assessment step where we have teacher agents whose job is to actually generate quizzes to see how well did the student agents actually learn those reference materials. And then if the student agents do not do very well on those quizzes, they have to go back to school, do additional readings, fine-tunings, model updates, and then retake these quizzes. So typical time for the agents to go through the school until graduate is usually about 24 hours or 1 or 2 days. So it's a little bit faster than human schools. Great. So now that we see how the agents can interact and also have gone through the schools, I'll come back to you and explain how the agents just go through their research project. And I will do this through this project, a concrete project that we looked at, which is how to designing binders to the recent COVID variants. Christian, did you have a question?
Yes. So you basically use the quizzes as performance measurements for when you're training the models, correct?
That's right, yes.
Okay, and the quizzes are they somehow evaluated before you post the quizzes? So is this some kind of real metrics behind that? Basically to measure the validity of the tests.
Yeah. Yeah. So here the quizzes are also created by other agents, right? So these are multiple choice quizzes, right? So I think the limitation of this is that these are all like agent design quizzes, right? So we also sometimes find it useful to have human input, right? So maybe have the human expert to say, okay, so test about these topics. But we mostly rely on the agents here to design these quizzes to make it more scalable because these agents are actually learning about topics beyond what I could teach it, right? Beyond things that we know about. So we want to have some more automated way of designing assessments.
So really a highly optimized approach also for setting up the performance measures.
Yes.
Yeah, very interesting. Thank you.
Great. Thanks for the questions. Okay, so this running case study, right? Recall that the agent's objectives to design binders, right, for these new COVID variants. And because these are new variants, right, so there are not really a lot of data for those, right? So how do agents overcome that data shortage challenge? Right. So to do that, the agents decided actually to create a computational model workflow, right, to simulate the performance of these different nanobodies and to the new COVID variants, right, to do that in silico to help it to optimize the design. So this is the new computational workflow that's created and designed, implemented by the agents. So first, the agents would actually use a tool called ESM, which is a protein language model. So ESM would actually take as input some amino acid sequence and then produce a score to say how likely is that sequence to form a stable protein. Right, so this would be sort of like the first filter that agent would use to look for stable proteins. And then they would actually take those stable nanobody proteins and pass that into AlphaFold together with the spike protein of the new COVID variant. So AlphaFold then generates this nice three-dimensional computational structure of how the nanobody candidate might bind to the COVID variant. So agent will actually look at that three-dimensional structure and extract some geometric information from that. And then the agent does not fully trust AlphaFold by itself, right? So then it actually also passes this AlphaFold structure into a physics simulator called Rosetta, which then would do more like molecular dynamics to sort of relax and then estimate the binding affinity. So then the agent also extracts the binding scores. Whereas these different methods or different tools all provide some complementary information. So the agents ends up creating like a holistic score by combining the output from these different tools. And then it would iterate through this pipeline multiple rounds to help it to select the most promising nanobody candidates. So I think this is actually quite interesting because this computational workflow is actually different from what humans have previously used and what people have published before. Right, so each of these tools is actually have been previously designed, been used, but the way of actually combining these different tools together is is different. So I think this illustrates a kind of the way that agents often work is through these more combinatorial creativity, whereby they can take existing tools and ideas, maybe combining that in a new way and to customize it to a new problem. And they can also make some tweaks and modifications to these existing tools. Right? For example, they actually took this ESM model. Previous papers had one way of doing the scoring metrics, right? So the agents actually developed a different scoring function to extract scores from this ESM model, which we found was also quite reasonable. So this workflow that will be created and implemented by the agents, and it was relatively fast at doing this, just within a few days. Right. And then by running this workflow, the agent's able to then come up with a small list, about 92 nanobody candidates, right? That the agents thought they were actually the most promising candidates. And this is also where the human AI interaction collaboration is critical, right? So then we would take these nanobody candidates, right? And then we, with our experimental collaborators, we made them, each of these nanobodies in the wet lab, we synthesize them, and then we test them in the wet lab to see how well do they work, right, as binders. So here I'm showing two example nanobodies. These are new proteins designed by the agent, so these are haven't been in existence before, right? And then the bottom curves show basically how well the experimental assays to quantify how well do they work as binders. So let me just explain what each of these curves mean one by one. If I look at the bottom red curve, right, was the open red circles. That's where we took previous human-designed nanobodies and test that against this new COVID variant. It's basically flat at 0, showing that they do not actually bind. The second red curve was a solid red circles. That's the AI-designed antibody. You can see that it goes up, so it shows more promising binding. And then the two blue curves shows where we take the same nanobodies and then test that against a different strain of COVID, right? The one from the Wuhan strain from 5 years ago. The AI designed nanobody, the virtual lab design one, which is a solid curve, also shows stronger binding compared to the previous human designed nanobodies. And I think this is useful because this shows that there's some signals, right, where the virtual lab design new nanobodies actually is able to bind across a broad spectrum of the virus. We also have additional experiments not shown here to show that if we take these nanobodies and test that against human host proteins or other viruses, they do not actually bind to our own proteins or our other viruses, which is also very useful as a way to reduce potential side effects. Right. So there definitely needs to be more experiments that we can do to further evaluate and quantify and optimize these nanobodies. But I think these results are already very promising to demonstrate that the virtual lab is able to, within a few days, right, design these new nanobodies, right, that we can then experimentally validate as being comparable and even better than some of the previous human-designed proteins. So just to summarize this first part, right? So we released this virtual lab platform. It's all open source as a part of our paper, and people can now use the virtual lab to tackle different kinds of research projects. So it's not limited to designing proteins. So you can use it to look for biomarkers, right? Analyze complex data and other applications. As a part of our study, we found that when you have teams of multiple agents with different expertise, right, that actually leads to better results than having a single agent to solve this problem by itself. Right? And the reason, as we discussed before, is that multiple agents often get into the discussions and debates that leads to, I think, to more creative and more robust reasoning and solutions. We also found it to be helpful to provide the agents with memories and resources, like a virtual sandbox, right? So the memory then serves as a digital notebook, so the agents have basically can remember exactly what they talked about before. And the local sandbox then provides each agent like a safe environment where they can locally modify tools like AlphaFold and run their own experiments. And finally, I think the agents have their own interesting social dynamics, right? So similar to how when humans collaborate, right? So, depends on people's personalities. That also leads to different trajectories of collaboration. Similarly, I think when the agents collaborate, they also have their own personalities. But something I think is really interesting is that we can also assign different personalities to different agents, right? So for example, we can make the critic agent more skeptical, or we can make the PI agent, more neurotic, right? And we can also see like what combination of personalities of these agents actually leads to maybe the most creative and most effective science. In that sense, then we can view the virtual lab as almost like a petri dish where we can do all of these science of science experiments and optimizations. That's would be very hard to do with human teams, right? But we can do that systematically with the AI teams, and then maybe take some of those insights to help us to inform how we can improve our own research groups. I'll pause here to see if people have any more questions about the virtual lab part before I move on to a second example.
Hi. Yeah, I have a question regarding the multiple simultaneous meetings. I think eventually they will converge, right? With so if you let multiple simultaneous meeting going on, they maybe converge to a few answers, or you would just how many simulations run you need.
Yeah, so they would usually converge, right? And it's actually also the job of the manager or the PI agent then to help these other agents converge, right? So the PI agent will often try to encourage other agents to reach some sort of consensus.
Hi, I have a quick question. I wonder if you have trained any agents with a more quantitative background, such as like statistical modeling, and are those type of agents like similarly like easily trained than a more scientific background agent?
Yeah, so it's a good question. So, for example, we do have like in this experiment, we have like a computational biology agent, right? So a little bit different from like a statistician agent, but the computational biology agent also is more quantitative, right? So it's actually the computational biologist agent that designed this computational pipeline and came up with the design of this pipeline, and then have the other agents help to implement this pipeline.
Okay, thanks.
Yes, I have another question.
Yes, I think Yvonne first, and then maybe Lydia.
So hi, I have a quick question about the AI teacher you're talking about. So I remember you mentioned like those knowledge of the AI are beyond like the knowledge that you have. So how do you know the AI teacher is giving the right assessment on those AI students?
Yeah.
Instead of like just judging them based on the wrong standards, so maybe they like pass without we knowing they're actually failing?
Yes, so it's a good question. So the teacher agents then basically would have access to, let's say, textbooks and reference materials that we think are high quality, like gold standard, right? So then they would design quizzes based on those textbooks and other reference materials. So that's how we sort of grounded the teacher agents. But ultimately, I would say like the final quality check comes from, here we actually do these wet lab experiments, right? So this is sort of the ultimate test to see can the agents actually then design something that actually works.
Oh, okay. So I have two questions actually. So the first one is, did you at any point try to compare maybe differences in the social dynamics like you mentioned, if it has any influence on the performance of the agent? That's the first one. Then the second question is, do you think this agent system would work in experimental settings?
Yeah. So the social dynamics we find to actually be quite important. For example, one experiment we did is that we removed the critic agent right, from some of these meetings. And we found that if you remove the critic agent, then the project ends up having a lot more mistakes, right? For example, a lot more coding errors later on. So that's where we found, okay, so it's really useful to have somebody who's more critical as a part of these initial discussions.
May I ask a question again, James?
Yes.
Since you're kind of from your experiments, multiple agents with diverse expertise definitely perform better than a single. And I imagine that would based on like expertise trained from some data or some training models. I wonder how could you detail in that aspect about how you did that? Do you use like your collaborators who carry those expertise particularly in each of their domains, or how do you get data to form those kinds of distinguished expertise to form such a team?
Yeah, so currently we do this through this agent school, which is a more of an automated process, right? And the expertise come from where the agents actually basically, for each area, right, the agents actually find the latest papers and reference materials. And then by reading from those and doing training and model updates from those material, that's how the agents is able to acquire more expertise in those specific domains.
Yeah, sounds great. Thank you very much.
Okay, great. So yeah, thank you for the good questions. So in the second part, then I want to talk about also follow-up work we had with the agents to the virtual lab. So if you view the virtual lab as sort of a way to reimagine what like a scientific team should look like, right? In the second part, there was paper to agents. Then we want to sort of reimagine how we disseminate knowledge, right, and what knowledge should look like. So, for the past hundreds of years, right, the way that scientists would try to share knowledge, right, and capture knowledge is in the form of these static papers. You can do years of experiments and you put all that insights into this paper. But from the other side, right, we think that the paper is actually a very inefficient way of sharing insights and knowledge, right? For example, there could be a lot of insights in how to run the code or how the data is collected that would be very hard for a reader to capture just by reading this paper or reading the PDF. So our idea is then, why don't we turn each of these passive artifact of knowledge, right, like these passive papers, into an interactive agent so that every paper then becomes its own agent. Right? So you can view the agent as sort of being like the virtual corresponding author that knows about the key details of that paper. And if the paper has any associated code, right, then this paper agent would also be an expert at using and running that code. Right. So in this case, right, instead of trying to like download the code from here and run it myself, which can take quite a bit of time, and then I can just ask the paper agent, right, the virtual corresponding author, right, can you help me set up the code and can you apply the tools from this paper to a new problem, right? And in this case, this paper agent then will just automatically do that, right? It will produce these reproducible workflows, generate these analysis traces, and come up with like the final solutions of how to apply this method to my new problem, right? So this paper agent, I think, then makes the process of like sharing insights and sharing code and data and research findings, I think, much more fluent. Okay. So the way that we actually do this is through this platform that we built called Paper to Agent, right? So Paper to Agent is actually an automated workflow that can actually convert a research paper into a corresponding paper agent. So here's sort of in a nutshell how this works, right? So we start with some original papers, and maybe there's some associated code base attached to that paper. We actually have a bunch of different worker agents, right? So these worker agents actually go in to try to understand the paper and code base. They will actually create like a virtual environment and try to simulate and reproduce results from the original code base and from the original paper. And by doing that iteratively, the outcome of these different worker agents is what we call a model context protocol, or MCP, that captures the key insights and know-hows from the original work. So these MCPs have recently emerged as sort of an industry standard way of capturing external resources for agents and for language models. Here, the MCP corresponding to this paper would capture all the tools, right, in this code base, insights from the manuscript itself, supplementary data, appendices, and also as well as like know-hows, like insights on how to actually use these tools effectively. So this paper MCP then is automatically generated as a part of our paper-to-agent workflow. And then we can then automatically host this paper MCP, right, on some remote server. And then by connecting your favorite language model and chatbot, right, to this paper MCP, this then enables the chatbot to become the virtual corresponding author that can actually run the code effectively and answer questions about the paper and then help readers, right, new users to apply the methods and data from the paper to new projects. So this is how we automatically convert a paper into an interactive dynamic paper agent. Right, so here's just an example of what this will look like. Right, so ScanPy is a pretty commonly used tool in bioinformatics analysis for single cell analysis. Right, so what the paper to agent would do is that it would read the original ScanPy and then go through the GitHub of ScanPy and then create the ScanPy MCP server, and then from that automatically create the ScanPy agent. Right, and then the users, like a new biologist, could just directly talk to the ScanPy agent and ask, can you help me to do my single cell analysis, right? And then here's my data, right? And then the ScanPy agent then would automatically use ScanPy and then generate the reproducible workflows and results and figures. And here, the way that we validate this ScanPy agent is to look at all these intermediate figures and analysis produced by the agent, and then compare that with analysis and results produced by a human expert user, right? That's done independently, right? And you can show that here there's a very an excellent match, right, between the agent-produced results and the human expert results. So we've done consistently across different challenging problems and benchmarkings to show that this paper-to-agent is actually able to answer these quite complex queries correctly. Whereas if you just simply, let's say, give these paper PDFs and code base, right, to Claude Code, right, or to ChatGPT or to other methods, then that actually leads to a lot more mistakes. Moreover, by going through the paper to agent, we can also reduce significantly the runtime, right, often by 2 or 3-fold compared to using Claude Code or using these simpler wrapper methods. So this demonstrates the benefit of first creating this robust paper MCP, right, and then building the agent on top of this paper MCP. So one part of this that I'm very excited about is I think this actually opens the door for many different new kinds of collaborations, including agent-to-agent collaborations. Right, so imagine that if somebody publishes a new methods paper, right, so then that gets automatically converted into a new method agent that knows how to use that method. Let's say a different research group publishes a new data paper, right, that also gets converted into an agent that knows how to use that data set. Then those two agents can actually get together and automatically collaborate and apply the method to the data and generate new analysis and new insights. Right. So this is actually a real example of this, right, where AlphaGenome, right, this very powerful foundation model from Google DeepMind, right, for doing sort of helping to predict effects of different mutations. Right, so with paper agent, we converted that into AlphaGenome agent. And then a different research group published a large-scale genetic study dataset look at risks for ADHD, and that's also automatically turned into a paper agent that knows about that dataset. In this case, these two agents actually got together and decided they could collaborate by applying AlphaGenome to help to interpret some of these GWAS results. And by doing that, they actually discovered a new splicing error that's significantly associated with increases in ADHD risk, which is not known before in literature. So kind of an interesting example of how now these different paper agents can directly collaborate. So just to summarize the second part here, right? So our vision here with paper agent is by converting all of these passive paper artifacts, right, into interactive agents, this can then greatly reduce the barrier to knowledge dissemination and make it easier to collaborate. Now it's much easier than for users to apply and adapt methods and data to new projects. This also enables a new form of collaboration, whereby maybe agents from different papers can directly collaborate, which in the past is often sort of bottlenecked by human communication limitations. But then here the agents do not have those bottlenecks, so they can directly collaborate and generate new results. I'll stop here a bit to see if people have any questions about the second part.
Actually, it's very interesting as well. Do you have a paper describing this approach, how to turn a paper into an agent?
Yes, yeah. So
Oh,
it's
so that's right there.
we have a preprint here. Yes, if you just Google paper to agent, you'll find our preprint doing this and also the code base.
Thank you.
A question about paper agents. I think you could probably also use smaller LLMs to represent a paper then, and so to enhance performance to save resources.
Yeah, yeah. So I think that's also interesting to explore. So here we mostly use Claude, right, to sort of the backbone for these paper agents because we found that the Claude is actually often very good at using code and applying these coding methods. But I think definitely interesting space to explore using smaller language models.
Thank you.
Okay.
Are you concerned about if you're over the agents in the sense becoming too constrained in how they, interpret, as it were, or convert the papers, and that you may lose an intuition that would have been otherwise been found because it's trained to be too rigid?
Yeah, I mean, I think in some sense, like the most rigid thing is the PDF itself, right? Because that's sort of passive, and then it's what you see on the paper is what you get. It's hard to really do something with that, right? I think that's often makes it hard for the readers to really understand what's going on. So here, the paper agent, we view that as sort of maybe a more flexible version of the original paper PDF, right? Because then you can talk to the paper agent, you can ask it questions, you can discuss with it, teach it things, right? And one version of this is that maybe even the paper authors themselves can also interact with the paper agent, right, of their own papers, and then teach those agents, right, how to better know some of these insights that were not captured in the original PDF.
Thank you.
Yeah, in the application base, let's say we're applying to a new data, the paper agent, do we need to like do a data preprocessing phase before implementing that, or would the paper agent do both?
Yeah, so it's a good question. So for example, with the ScanPy agent, right? So there, it will actually do the pre-processing of the single cell data for you as a part of, so it knows how to do that. I think some of that depends on like how, if the data is relatively standardized or common kinds of data, then the agent should be able to do that.
Good. Then another thing, if we have some packages that have been retracted from maybe a previous paper and we're trying to use the paper agent to reproduce, how does the paper agent address that?
Yeah, so it's a good comment. So I would say that not all the papers and not all the code bases can be agentified fully, right? So for example, if a code base is missing some key code, right, or it doesn't even share the code, right, or it has mistakes, then in the process of trying to create an agent from that, right, the paper agent will actually raise different warning flags and say, oh, this part of the code base is not reproducible, or we cannot actually create an agent from this. Right. And that does happen. And we think that that itself is actually a useful signal to the community, right? Because if a paper maybe cannot be fully identified, that probably means that maybe there are some gaps in the paper or in the writing that should be filled in.
Thank you.
Great. So maybe I'll just more briefly go through the last part in a couple, in 5 minutes. So we have a bit more time for some additional questions. So what we did here is that we actually put all of these different pieces together, right? And we organized this conference we call Agents for Science. It's an experiment, right? The conference is the first conference where both the authors and also the reviewers are AI agents. And the motivation for organizing this experimental conference, right, is that there are a lot of questions in the field, right, about how creative are these AI scientist agents? What's the best way for human and AI to collaborate? Can AI review papers? And it's actually very difficult to answer these questions systematically in a data-driven way because the existing conferences and journals do not allow AI as co-authors or as co-reviewers. Right, so we thought, okay, let's flip this around, right? Let's create this new conference where you have to be an AI to submit to the conference. Right? So humans can be co-authors, but AI needs to be like the first author. And then every submission can also be reviewed by other AI reviewers, right? So that's, and importantly, we make all the submissions and reviews publicly available. So then this becomes a community resource so that we can all learn about what are the strengths and limitations of AI scientists. Every submission also, we require documentations of how the AI and humans collaborate. So we can learn about best human collaboration patterns. And then we also have human expert assessments for a subset of the top papers. So then you can compare the human reviews with the AI reviews. So we we ran this conference last year in last October. We received over 300 submissions, right, from 28 different countries. From this, 48 papers were accepted right after these AI reviews. So these submissions covers quite diverse domains, right, including applications of AI in physical sciences, in life science, and social sciences. People also use quite different models, including GPT, Gemini, Claude, but also some of these more open source models. So maybe the main one thing that's quite interesting is the collaboration patterns. For every submission, we require a detailed documentation of how did the AI and human collaborate across each of the steps of research, right? From developing hypotheses to designing experiments, data analysis, and even the writing of the paper itself. So first, let's look at all the submissions here on the left. Right, so red basically means that this is done essentially all by AI, right? Green means that it's done by human, right? Orange means that it's done mostly by AI with human feedback, and blue means that it's mostly human with AI feedback. So you can see here that a good number of these submissions are where the AI actually came up with the idea, right? These are red ones, right? And AI also do the experiments, data analysis, and writing. So these are fully autonomous papers. We do see a trend whereby there tends to be more human involvement relatively in the earlier steps of research, like hypothesis development and experimental design, and then more full AI autonomy, right, in the later stages, like data analysis and writing. It's also interesting to compare all the submissions with all the accepted papers, right? Here we found that, for example, the red bars here, right, the full autonomous AI is less in the accepted papers than compared to the overall submissions. So that suggests that at least among the more higher quality papers, there still needs to be some human involvement, right? Not just fully autonomous AI in order to guide and to ensure quality. So we have more details of this analysis published in this paper here, where we summarize the learnings from this conference. One interesting learning is that we also used AI reviewers. Right, so basically, every submission, we had 3 different AI reviewers that we calibrated from using data from previous conferences to review. Right, then we chose these reviewers based on 3 popular families of models, so GPT-5, Gemini, and Claude. And even though all 3 reviewers were given the same instructions and reviewing the same papers, we actually found that they actually often give very different scores. So they have very different personalities, right? So for example, GPT-5 is consistently maybe the most negative of the reviewers, right? So its scores tends to be low. Gemini is at the other extreme. It tends to be the most positive of the reviewers. Often for a large number of papers, they actually give a very high score. And Claude, we think it's more balanced. So in between these two, and maybe more aligned with human expert reviewers. Here we found that the AI reviewers are quite good at catching often mistakes and inconsistencies in the paper. For example, here it finds that there's some inconsistencies between the statistics reported in the text versus in the table. There are also some limitations for these AI reviewers. One limitation is that we found that some the reviews, especially from Gemini, tends to be too sycophantic, right? For example, the Gemini reviewer would say that this is a groundbreaking study, but the human assessment was more skeptical, right? So sometimes the AI reviewers because of the way they're trained, maybe tends to be wants to please the human users too much. People are quite interested in how much do these papers hallucinate. And the way we do this is that we can actually have a separate system that takes every reference in these submissions and then search and see if this is actually a real reference or not. So each dot here is one of the submitted papers. The x-axis, the total number of references, and then y-axis is the number of references that we're able to independently verify. So you can see that maybe about half of these papers are essentially on the diagonal, right? So there are all of their references are actually real references, but about 50% of these submissions had at least one hallucinated reference. And some of these dots near the bottom here in red are these papers where most of the references are all hallucinated. Right? So this shows that hallucination is still a relatively widespread challenge with these AI-led research papers. Finally, here's just one example of a submission to the conference. This is actually a social science application, right, where the AI agent with a few human collaborators, right, decided to simulate job markets where both the companies that are looking to hire and also the people who are looking for jobs are actually AI agents on both sides. And they use this as a simulation platform to quantify the effects of different job policies. So this was actually also reviewed by human experts who's a Nobel laureate in economics. And in their review from the human experts, the expert wrote that this is actually a really interesting paper. It's technically well done, right? And it's also like a very fascinating topic to using agents to simulate both sides of the job markets. So I think this is one good example where, according to a very high level of human expert, right, that they thought that this is actually the AI is able to do something quite creative and making an interesting contribution to the field. So maybe in the interest of time, I'll just wrap up here. Right. So I wanted to show these three examples to illustrate some of the promising developments of AI agents for science. Right. So AI scientists, not just as a tool for science. Right. In virtual lab, we see how teams of AI agents can collaborate to tackle open-ended problems, and they actually discover new nanobodies that we experimentally validated. In paper to agent, we can see how by converting passive paper artifacts, right, into interactive dynamic agents, like virtual corresponding authors, that this can facilitate collaborations. And as Agents for Science, this sort of like experimental first of its kind conference where both the authors and reviewers are AI agents. And we saw some quite interesting research and innovations done by the agents here. I want to especially thank my students that led these different projects. So Kyle Swanson for the virtual lab, Chad Chimmel for paper to agent, Federico Bianchi and Owen Quinn that led the Agents for Science conference. And these are the references for each of these works. Thank you.
Thanks, James,
Thank
for
you, Dr.
a
James.
fascinating talk. Yeah.
Thank you.
So we have time for a few questions. People want to raise their hand. That's the best way. Yeah, Abir, go ahead. Sorry.
Thank you so much. Very interesting and fascinating talk. I'm really curious about your budget for conducting or running an entire conference to test the peer review and the submission process. Was this funded by some agency?
It's a good question. So we basically used my sort of discretionary funds for this. Because we weren't sure how like standard agencies will feel about this experimental. Yeah. So the budget for this, I think we spent maybe a couple $1,000 for the AI reviewers, right, for training and applying these different AI reviewers. But the rest of it is actually relatively inexpensive.
So it was a virtual conference, not in.
Yes, and the conference itself is a virtual conference. And the recording of the conference is all available on our website. If you search for Agents for Science, you can find the recordings of the conference as well as all the submissions and reviews.
Yeah, that would be impressive to look at. Thank you.
Any other questions for James?
So what would be the suggestions for us as researchers? moving forward, what are the good things we could do? What are the things we should avoid?
Yeah, I think it's definitely a very rapidly changing field, right? Under these new ideas, new technologies coming up. I do think that these AI agents, if they're used appropriately, right, with human guidance, can help us to accelerate research quite a bit. But we also do not want to become necessarily like overly reliant on these AI agents, especially, for example, like in when we train the students in classes or in our research groups. I think there are still parts of research where it's important for the students to be able to do themselves, even if the agent can do it faster. Right? Just I think it's important for the people to learn how to do that in a hands-on way.
Thank you very much.
Great.
Great.
Well, if there are no other questions, then well, thank you very much, everyone. Enjoyed the discussions.